So today we'll discuss cell cycle and cell division. So all we know, our body is made up of different types of cells, number of cells. So cell division is an inherent property of living organism. It is a process in which cells reproduce their own kind. Okay, cell division means it is a process in which cells reproduce their own kind, means that cell shows or produces new cells as like their own cells. The growth, differentiation, reproduction, and repair take place through cell division. Okay, means our cell, that cell growth, then cell differentiation, then reproduction and repairing of cell, that occurs by the process of cell division. In the cell division, there are two types of cell division, which is namely mitosis and meiosis. My voice is clear?
Yes, sir.
Yes, sir.
Okay. So cell division, which consists of two types, that is mitosis and meiosis. And the cell which is capable of undergoing division process, that division which is occurs through cell cycle. Okay, means there is one cycle, there is one event cycle occurs during cell division. Okay, so what is this, or what are the various steps we will discuss? Okay, so next we'll discuss about cell cycle. Means through this cell cycle, cell division occurs. So there is some of the sequence of events occurs during that cycle. The cell cycle is the sequence of events or changes that occur between the formation of cell and its division into daughter cells. Okay, means through this cell division, that cell cycle, which consists of series of events or series of changes that occurs between the formation of new cell, between the formation of its daughter cells. Then the cell cycle has non-dividing growing phase, that is called as interphase, and it consists of dividing phase, that is called as mitotic phase or M phase. Means that cell cycle, mainly it has two phases. First phase is non-dividing phase, or which is known as growing phase, which is known as G phase, and second, which is a dividing phase, which is called as mitotic phase or M phase. Okay, so cell cycle, which mainly divided into two phases. G phase means growing phase, second M phase means mitotic phase. So what is that? We will go for that sequential events. What sequential events occurs in that G phase and M phase? So G phase, it consists of again three subdivisions. in that G1 phase, S phase, and G2 phase. While M phase, here a cell division may occur. So here mitotic phase, it is a dividing phase. So now we will discuss one by one. So in that, first is that interphase, and second is M phase. Interphase, nothing but the growing phase. Okay, so inter means between and phase means aspect. So between the aspects or growing phase, nothing but the interphase. So interphase, okay, main cell cycle have two phases, interphase and M phase or growing phase. Interphase, nothing but the growing phase. Second, M phase is nothing but mitotic phase. Now we will discuss in detail what is growing phase. So first, interphase. So inter means between and phases means aspects. So it is a long metabolically active phase between two successive mitotic cell division. It has again three substages. Means during growing phase, that cell cycle interphase, which is having three subdivisions. So in that G1 phase, S phase, and G2 phase. So now we will discuss interphase. So interphase, which is subdivided into three: G1 phase, that is first growth phase, then S phase, which is known as synthetic phase, and third is G2 phase, that is known as second growth phase. So G1 phase, what events occurs during G1? Interphase means growing. In that, cells becomes grow. Okay, simply growing means that cells growing in the interphase. So first G1, it is the first growth phase. What happens in first growth phase? The cells prepare for DNA, RNA, and protein synthesis. During G1 phase, that cells which prepares their own DNA, RNA, and protein synthesis. Then second subphase, S phase, that is known as synthetic phase. So during the synthetic phase, the duplication or replication of DNA and centriole takes place during the synthetic phase or S phase. Then third subphase, that is G2 phase, that is known as second growth phase. In that, the synthesis of proteins, which is required for synthesis of spindle fibers, takes place in the G2 phase. So this is about interphase. Okay, you can see the previous slide. So look at this. First is that G1, first growth phase. So in the first growth phase, DNA, RNA synthesis occurs. Then during S phase, replication of DNA and RNA occurs. And G2 phase, in that G2 phase is mainly responsible for preparation of mitosis. That is again synthesis of proteins, which is required for the synthesis of spindle fibers, takes place. So that is about interphase. Now we will go for M phase or mitotic phase. So M phase or mitotic phase, it is a short phase, and again it includes two processes simultaneously, that is karyokinesis and cytokinosis. Mitotic phase, which again divides into two groups, karyokinesis and cytokinesis. Karyokinesis means division of nucleus, and cytokinesis means division of cytoplasm. Okay, means our M phase, which is divided into two subdivisions, karyokinesis division of nucleus and cytokinesis means division of cytoplasm. And by division of that cytoplasm, it results into two daughter cells. Okay, means two new cells formed after that M phase. So after M phase, the cell may enter either interphase to repeat the cell cycle, or goes to the G0 phase to arrest cell cycle. Okay, means after completion of M phase, that cell cycle either goes to the again new daughter cells development, that is interphase, and sometimes death of cell occurs, that is it goes to the G0 phase. So cells in G0 phase may grow and differentiate into two cell types to perform different function. Okay, so this is interphase and mitosis phase. Now we will discuss about what is mitosis phase in deep. Up to this, do you understand? इथपर्यंत लक्षात येते का?
Yes, sir. Yes, sir.
Okay. So now we will go for the next. What is M phase? What event occurs in M phase? What are the changes occurs in M phase? So mitosis phase, M phase nothing but the mitosis. So this is a continuous process which involves 4 distinct stages visible by light microscopy. Okay, so that mitotic phase, which again subdivided into 4 stages. For that purpose, you can remember PMAT. Okay, PMAT. So mitotic phase divides into 4 subdivisions, 4 distinct stages. So in that, first is known as prophase. Then second is that metaphase. Third, anaphase, and fourth, telophase. So there are some changes occurs in the cell or dividation of cell. Mitosis means main dividation stage. So first of all, prophase. So prophase, during this stage, the replicated chromatin becomes tightly coiled and easier to see under microscope, and each of an original 46 chromosomes, which is called chromatid at this stage, is paired with its copy in double chromosome unit. Means formation of 2 sets of chromosomes. How? The true chromatids are joined to each other at the centromere. So inside the cell, we have centromeres. Okay. So during study of cell, we have discussed about cell. What is centromere? So 2 chromatids which is joined to each other at the centromere. The mitotic apparatus appears. This consists of 2 centrioles separated by mitotic spindle, which is formed from microtubules. After that, that centromers which goes to the separation. Okay. As I have discussed, a mitotic division which occurs by nucleus division, and second thing is that cytoplasm division. Okay. So again, the nucleus which is divided as well as cytoplasm is divided. But during that, the two centromers which goes to the opposite directions for dividation purpose. And after that, the mitotic spindles which are separates that two centrioles, and that mitotic spindles which is formed by microtubules. Microtubules nothing but the tiny protein fibers which is present in our cell, as we have discussed in cytoskeleton. After that, in the prophase, the centriole migrates one to each end of the cell and the nuclear envelope disappears. What happens means that two centrioles which goes each end of cells means goes to the opposite direction, and through this nuclear envelope becomes disappear. Okay, in the cell, the nuclear envelope becomes disappear during that prophase. So this is about phase 1, prophase. Now second phase is that metaphase. So during metaphase, the chromatids align on center of the spindle and attached by their centromers. Whatever the chromatids which is present in nucleus, that is DNA, that chromatid which again aligned on the center of spindle attached by their centromers. Then phase number 3, anaphase. During phase 3, anaphase, the centromers separate and one of each pair of sister chromatids, so that sister chromatids which migrates to each end of spindle as microtubules, that forms the mitotic spindle contract. So that part we will better understand in figure. Okay, in next slide we have a figure, and that point we will get better understanding. So that is phase number 3, anaphase, that separation of spindle fibers at each end. Now fourth phase, that is telophase. The mitotic spindle disappears and chromosomes uncoiled, the nuclear envelope reforms. And that following telophase, cytokinosis occurs. Means after the during that telophase, again dividation of cytoplasm. In the prophase, that is dividation of nucleus, but in case of telophase, cytoplasm becomes cytoplasm dividation may occurs, that is known as cytokinosis occurs. That is cytosol, intracellular organelle, as well as plasma membrane, which splits forming two identical daughter cells, and after that, that material of cytoplasm divides into two daughter cells. So for that better understanding, we will go for the next slide. Here we can understand the each phase. So look at the image, look at the stages of mitotic division. So new slide is visible, stages of mitosis and cell division. New slide is visible?
Yes, sir.
Okay, look at that.
Yes, sir.
So first of all,
Yes, sir.
interphase. So during interphase, so it consists of 3 substages. What are the 3 substages of interphase?
G1 phase, synthesis phase, and G2 phase.
Yes, correct. G1 phase, synthesis phase, and G2 phase. So that first is that interphase. After completion of interphase, So dividation phase may starts, that is we called mitosis phase. So in that first phase is prophase. So during the prophase, that chromatids becomes separated and centromers which align on opposite directions. So that part we can see in the late prophase. So that centromers goes opposite directions. So white colored, that is centromer. And through this, after that late prophase, it goes to the metaphase. So centromers makes chromatid alignment at the center in the metaphase. You can see the chromatids, that XX, which is the DNA or chromatids, which aligned at the center, which binds towards centromer. From metaphase to anaphase, the separation of chromatid, separation of that DNAs, that is spindle formation. Spindle becomes separated in anaphase. And last, you can see telophase. Again, separation of cytoplasm, that cell which is divided two equal parts, that is telophase. And through this, it forms cytokinosis, that is the formation of two daughter cells. After that, it forms two daughter cells. And this is about stages of mitosis and cell division. So today we have to discuss that part, cell cycle and cell division. So cell cycle, which is divided into two phases, mainly, that is interphase and M phase. Interphase nothing but the growing phase. M phase, mitotic phase. So interphase, which subdivides into three phase, that is G1 phase, S phase, and G2 phase. M phase, which divides karyokinesis and cytokinosis, that is prophase, metaphase, anaphase, and telophase. So during that, the dividation of cell occurs into two daughter cells. Okay, so this is about stages of mitosis and cell division. Do you understand? समजते का सगळ्यांना?
Yes, sir.
Yes, sir. Yes, sir.